Earlier, we have discussed how histone tails can be post-translationally modified to alter chromatin compaction and access to the DNA. This video provides an introduction to histone acetyltransferases, or HATs, the enzymes that add acetyl groups to histones. As we have discussed previously for other marks, the mechanisms governing epigenetics marks such as histone acetylation can be summarized by the writer, eraser, and reader model. For each mark, we typically have writer molecules that add the mark to the chromatin, and we have eraser molecules that can take the mark off again. In addition, we have reader molecules that can determine if a mark is present or not and lead to a specific outcome, gene expression versus gene silencing, for example, depending on the presence or absence of the mark. For histone acetylation, the writer proteins are histone acetyltransferases, the eraser molecules are histone deacetylases, and the reader molecules are bromodomain-containing proteins. Here, we will focus on the writers, the histone acetyltransferases. Histone acetyltransferases, or HATs, are simply enzymes that can transfer an acetyl group from a donor molecule to a histone protein. In some cases, despite their name, these enzymes also have non-histone targets. The addition of acetyl groups to the histones leads to the reduction of their positive charge. Thus, it reduces the strength of the interaction of the DNA with the histone proteins and makes the DNA generally more accessible. In the figure on the right, you can see the molecular structure of a histone acetyltransferase from Tetrahymena, GCN5, in complex with the acetyl donor, acetyl-CoA, and the histone peptide that will be acetylated. Not surprisingly, given the large number of acetylation sites on the 4 core histones, there are a large number of different HATs. The diagram on this slide shows a number of HATs from the yeast Saccharomyces cerevisiae, their names begin with Y, and from humans, their names begin with an H. All HATs have a HAT domain, in red, which is the catalytic domain that actually moves the acetyl group from the donor molecule to the histone protein. Many HATs have additional conserved domains such as bromodomains, chromodomains, or zinc finger domains that help localize them to target sites in the genome or mediate interaction with other proteins. HATs are typically classified into different subfamilies depending on their exact mode of action, meaning how they move the acetyl groups from the donor molecule to the histone molecule. Shown on this slide are the 5 major subfamilies of HATs. They are the HAT1 family, the GCN5 PCAF family, the MIST family, the P300 CBP family, and the RTT109 family. A general feature of HATs is that they require acetyl-CoA as cofactor and donor of the acetyl group. HATs are typically part of larger complexes, such as the SAGO complex or PCAF complex shown to the right. Other proteins in the complex typically regulate the function of the HAT, its specificity to a particular protein target, and its activity. HATs also often participate in more than one complex, and thus can have different target specificities. In addition, several of the known HATs control their activity by autophosphorylation. Given that the addition of acetyl groups to histones leads to a weakening of the interactions between the DNA and the histone proteins and makes the DNA more accessible, it is not surprising that HATs have many important roles. HATs are important generally for gene regulation and more specifically for cell cycle progression, DNA repair, hormone signaling, and dosage compensation. They also play significant roles in human diseases, including a variety of cancers, but also in acute infections and chronic diseases such as diabetes. Because of their roles in various diseases, hats are under investigation as possible drug targets. A variety of HAT inhibitors have been investigated, including curcumin, a compound that is found in curry powders and turmeric and is sold as supplement. While these inhibitors are promising drugs, many suffer from moderate potency, meaning that their inhibitory effect is not very strong. In addition, because there are a large number of HATs which all share a very conserved catalytic domain, many of these inhibitors show only moderate specificity and inhibit more than one HAT. In summary, in this video, we have explored how histone acetylation is produced by HATs. Histone acetylation is a post-translational modification of histones that weakens the interaction between DNA and histones. HATs are divider molecules producing histone acetylation. There are 5 different subfamilies of HATs, and HATs in all subfamilies need acetyl-CoA as cofactor and typically function in large complexes. HATs contribute to many cellular processes and are involved in several diseases. And due to this contribution to disease, HAT inhibitors are being explored for medicinal use.